We'll talk about T cells first, and the differentiation of T cells. Do you remember where this took place again? Where did T cells? What starts with a T? Thyroid? No. It does start with a TH, so it's thymus. Yes, thymus. But they're not made in the thymus. No. Just like every other type of blood cell or hematologic cell, they are made in the bone marrow. Okay? But then they travel to the thymus. There we see the thymus in the... Where is it located again? The anterior mediastinum. Travel to the thymus. And at this point, they are undifferentiated. There's two main types of T cells, either CD, and CD stands for cluster of differentiation. That's the receptor that is on cells. You'll see a bunch of different types of CDs, okay, clusters of differentiation, but that is a cell's unique receptor. So they can either be CD4 or CD8. CD8 cells become cytotoxic T cells like we just discussed. They kill virally infected cells and tumor cells. CD4 cells become helper T cells. Okay? The way I remember that is CD8, if you ate something, that means you consumed it and you killed it. That's what CD8 cells do. They are cytotoxic. They kill cells. Okay? It's a good way to remember it. So, what happens first, and remember, where is this taking place in the thymus? Starts in the cortex on the outside, right? That's where they start differentiated. So what happens first is positive selection. The thymus makes sure that these T cells are able to bind to foreign antigens. So they have sample foreign antigens like pieces of bacteria or some other pathogens, some other PAMPs, and they make sure that the T cells can bind to them. If they don't bind, they don't complex, then they are apoptosed and destroyed. Okay? That's called positive selection. The next type of selection that occurs deeper, in what part of the thymus? In the medulla, is negative selection, okay? Negative selection. So this is where the body actually takes native cells and says, okay, T cell, are you going to bind to this? Yes or no? If you attack your own cell, the own body's cells, then you should be killed. You should be apoptosed. So that's called negative selection. If they destroy their own cells, they're apoptosed. And we're dealing with specifically CD8 T cells because CD4s are just helpers. CD8, remember, 8 cells, it's cytotoxic. Okay, so MHC receptors. What do CD8 cells bind to? We just said they're very analogous to natural killer cells because they bind to, what type of MHC is this? MHC1 receptors on the surface of virally infected and neoplastic cells or cancer cells and cause them to apoptose. So a way to remember that is the multiple of 8 times 1 is 8. And we're going to see that CD4 are going to complex with MHC2 receptors, and 4 times 2 is 8. So the way you remember what cells bind to what is that the product is always 8, okay? It's going to help you. So like I said, they bind to virally infected and tumor cells and cause those infected cells to apoptose. Very similar to what we saw with the natural killer cells. So that's CD8. And like I just said, CD4, what type of MHC will they bind to? 4 times what is 8? 2. It's MHC2. And where do we see MHC2 receptors? Where are they? On antigen-presenting cells, right? Like macrophages, B cells even, and dendritic cells. Now, CD4 cells themselves can split off into different types. So, remember, CD8, what type of cell is that called? Cytotoxic, because it ate cells, right? CD4, what type of cell? Helper T cells, right? Because it helps out, releases cytokines. But there's different types of helper T cells. So let's talk about them. So depending on what cytokines these helper T cells are exposed to, they differentiate into a specific type of helper T cell. So if they are exposed to interleukin 12, then they will turn into Th1 type cell. So T helper 1 type cell, all right? And T helper 1 cells, what do? Well, they release cytokines like most helper T cells do, but what types of cells do they stimulate? B cells. They help B cells class switch and change from making IgM into IgG and other types of immunoglobulins. And these cytokines stimulate macrophages and other antigen-presenting cells to process antigen. All right? So that's what Th1 cells do. And the way I remember that interleukin 12 stimulates the formation of Th1 cells is that the 1 is first. The number 1 comes first, and that's what cell it becomes. Okay, so 1 is before 2. All right, so I should mention too that they also stimulate, as we see here, CD8 cells as well. So Th1 cells are just the big helpers. They help everybody. If this undifferentiated CD4 cell is given interleukin 4, it will become a T helper 2 cell. And these are a little bit different. They are important in allergy. Okay, so here's a B cell. And like we said before, B cells make what immunoglobulin that gets stuck onto the surface of mast cells? IgE. IgE, right? So IgE, stuck on the surface of mast cells, exposed to an antigen or an allergen, cross-links the mast cell, causes degranulation of the mast cell's contents, and massive allergic reactions. So you could think of interleukin 4 causing allergic reactions, all right? And Th2 cells, think Th2, achoo. That's kind of how I remember it because it's associated with allergies. All right, and finally, there is one other type of CD4 cell. When exposed to TGF-beta, it will turn into, well, do you know, first of all, what type of cytokine TGF-beta is? Transforming growth factor beta. That is an inhibitory cytokine. So it makes sense then that this is going to turn into an inhibitory type of T cell. So it's a T regulatory cell. All right? And this just goes and inhibits things, pretty much all types of T cells. Tells them to stop, because everything needs to be told to stop sometimes. Flash quiz. Time for flash quiz. Woohoo! All right, hopefully it's easy again. Although both arise from the bone marrow, where do B and T cells mature? So, bone marrow for B cells and thymus for T cells. It's easy enough, right? And you knew that. The B cells stay in the bone marrow to keep maturing. T cells travel to the thymus. Nice alliteration there. Okay, so by now you're getting a pretty good idea of what helper T cells do. And remember, there are two main types of helper T cells. So let's talk about those two main types in a little more detail just to clarify and completely solidify in your head what types of T cells there are. All right, so there's Th1, and we said again, Th1 cells, what do? They stimulate macrophages to process antigen. And we already talked about the B7 and CD28 receptor. We just mentioned all of this stuff. Tumor necrosis factor alpha and interferon gamma, co-stimulation, cytokines, more stimulation of other cells, etc. Yep, you know that. This is just a review. Th2, we didn't cover in detail yet, but you have a great idea. So here's a B cell, and it's making IgM. And we just talked about that Th2 cells, they can stimulate B cells to make IgE instead of IgM, and there are specific interleukins, and those are actually interleukin 4 and 5 that will stimulate B cells to make IgE. So you just have to know interleukin 4 and 5 make IgE. And remember, actually, what it was special about interleukin 4. What did it actually cause the differentiation of? It told the undifferentiated CD4 cells to become Th2 cells. So just like it stimulates these Th2 cells, it also stimulates B cells to make IgE. So interleukin 4, think allergy. And remember, IgE on the surface of mast cells, cross-linking of allergen, release of granules, and allergic response. Okay, so now that we just dealt with the helper T cells, the CD4 cells, now it's time to talk about the CD8 cells. The cells that ate other cells. They're cytotoxic, remember? Here we have, remember, 8 times 1 equals 8, so they complex with MHC1 receptors. And these are typically cells that have fragments of viruses or tumors presented on their surface. So, CD8 cells complex, and now we're going to see what are these receptors here, huh? What do you think? Fast ligand is on the T cell, is on the CD8 T cell. All right, know that, and that complexes with CD, do you know? 95, that's the fast receptor. And this is the so-called death domain, because once this first step occurs, all it takes is a cytokine to stimulate this poor cell to kill itself. What cytokine is that, and what's it released from? Remember this helper T cell Th1 type? So this is Th1. This helps all kinds of cells, including inducing apoptosis. That's interleukin 2. It uses interleukin 2 actually to stimulate the CD8 T cell to put the dagger, the nail in the coffin inside this cell. Remember what's released from the mitochondrion relating to apoptosis? Cytochrome C. Cytochrome C then goes and activates caspase enzymes. Voila, apoptosis. A nice, neat, organized cell death. So just don't get confused. Just break down your T cells into, when you're thinking about them, into are they helper ones? Are they CD4 or are they CD8? Okay, and you immediately think 8 as eating cells. It ate something that's cytotoxic. That kills what types of cells? Virally infected cells and tumor cells. CD4, well, what types of cells do those stimulate? If it is, we break it down into what? They're both helper, but there's helper type 1 and helper type 2. If it's helper type 1, it pretty much stimulates everything, except for allergic response. If it's Th type 2, you think of a chew. And that's the type of T cell that stimulates IgE formation and the allergic response. So that's how you break down your T cells. Just do it logically. Think about 4 versus 8, and then you know what MHCs these complex with. Remember, CD8 must complex with MHC1. CD4 must complex with what? MHC2. Very good. So you got it. It's easy. Just keep that in mind, and you can't get lost. And the cytokines and the smaller details and the receptors, those will come along the way. Just keep reviewing your stuff. So, also, I should mention that undifferentiated CD4 cells can turn into regulatory T cells as well. Remember the cytokine involved with that? TGF-beta, right? Right. What cytokine caused the formation of Th2 cells? Interleukin 4. Very good. What about Th1 cells? Interleukin 12, because 1 is before 2. Good. So you got it down. You're getting it down. All right, so regulatory T cells, what do they do? Well, like I said, they are just inhibitory. They regulate things. And interleukin 10 is a big regulatory interleukin. So is TGF-beta. So oftentimes, like we saw with interleukin 4, the cytokines that actually stimulated and caused the differentiation of these T cells, they are usually produced by the type of T cell and cause the downstream effects of that T cell. So this one is responsible for releasing TGF-beta, the same thing that caused its differentiation into a regulatory T cell. Just like Th2 cells release interleukin 4, and they were differentiated by interleukin 4. Just know, too, that the receptors on regulatory T cells are C3, 4, and 25. That could be asked. You'll get your 99th percentile in the test if you know that. Let's now delve into some more of the nitty-gritty details of how T and B cells get activated. It's going to be fun. So, here's a macrophage. All right, and we're going to talk about the specific receptors. And in general, there are always 2 main signals that need to occur for an immune cell to be activated. All right? 2 signals, at least. In addition to the initial receptor. So just remember that. So, keeping that in mind, here's a macrophage. And again, what type of MHC is this? MHC2. So this macrophage, let's say it ate a bacterium, processed it, places it on its surface, on its MHC2, and presents it to, again, what type of T cell will complex with an MHC2? What times 2 equals 8? 4. So this must be a CD4, and do you think this is a CD4 helper type 1 or 2? Well, remember, 2 was achoo, that was allergy, so this must be CD helper type 1. Right? Right. Remember, the Th1 cells, they really help just about every type of cell. So if you're really in doubt, you could probably go for Th1. So CD4 T helper type 1 complexes with this macrophage. And now let's see if you know the specific types of receptors that are involved here. So there's B7 on the macrophage, and that complexes with what on the T cell? I'll give you a hint. 7 times the CD4 complex, the CD4 number, equals this T cell's receptor. You'll see what I mean. So 7 times 4 is 28. So CD28. That's how you remember it. Make sense? Good. And so that's the first signal. That was signal number 1. All right? And so now, usually the second signal involves cytokines. And macrophages release tumor necrosis factor alpha. And the way I remember that, it's kind of silly, but macrophages, the second letter is an A, which makes me think of alpha. So macrophages. It kind of helps. I don't know if it helps you or not. So anyway, TNF alpha goes and stimulates the T cell, and there's actually co-stimulation. The T cell will release something. What cytokine will the T cell release? Interferon gamma. And I think of the gamma kind of looks like a little bit of a bent T. That's how I remember that the T cell releases it. So the interferon gamma goes and in turn stimulates the macrophage, and then thus they both co-stimulate each other. So that is the second signal. So we had 2 signals. Now we have full activation of these immune cells. Great. And now they can go and do their thing. The macrophage will process antigen fully and release more cytokines and get other cells going. The T cell will release other cytokines and motivate other cells. So that's what will occur once you have co-stimulation. Very good. Now let's talk about B cells. So how do they get activated? Again, 2 signals need to occur. So here's a B cell. And just so you know, B cells have IgM and IgD on their surface, type of immunoglobulins. All mature B cells have that. You can think MDs are mature, IgM and IgD. Usually doctors are mature, so you can think MDs are mature. So they also have what type of MHC? MHC2 receptor. And let's figure out what this receptor is. Woohoo! Exciting, right? Let's say they have a fragment of pathogen on their MHC2 receptor. They're going to present it to what type of T cell? 2 times what equals 8? CD4. And what type of CD4 cell? Well, in this case, it could be either a type 1 or 2. Because a type 2 could also stimulate the B cell to make IgE, right? But let's say this is a type 1 again. So Th1. And let's now talk about, do you know what receptors these are here? CD40 actually is on the B cell, and then what is on the T cell? CD40 ligand. So there's the first signal, all right? Signal number 1 for co-stimulation is just taken place. So exciting. All right, so that causes, then usually the second signal, like I said, is involved in cytokines. So various cytokines, we'll go into what they are specifically later, but various cytokines and types of interleukins then will stimulate the B cell to class switch. And if it was making IgM, then it will make, well, maybe IgG, maybe IgD, maybe IgA, who knows? Any type of immunoglobulin after IgM. So that's what happens with B and T cell stimulation. Time for a flash quiz. What do B cells synthesize and release? So easy. Come on, guys. Just to keep you awake. Antibodies. Very, very good. And what's the first one that they synthesize? IgM. That's the immediate one.